Bonjour,
Bonjour.
Hélène. Donc Hélène
Bonjour.
vient de l'Institut Pasteur et donc on continue avec la recombinaison homologue.
Oui, c'est ça.
Et voilà, on reste un peu dans le même thème.
Je partage mon écran. Hop.
Normalement, tu dois pouvoir. Voilà, c'est parti. Super.
Alors, attendez, ça ne marche pas. Ah, si, c'est bon. C'est bon?
Oui, ouais, impeccable. Donc,
Ok.
on est parti donc, pour 20 minutes à peu près. Je te préviendrai un peu à l'avance. Oui.
Ouais, d'accord. Ok. Donc, tout d'abord, bonjour à tous. Donc, tout d'abord, je voulais m'excuser parce que je vais faire cette présentation finalement en anglais. So I would like to thank the member, of course, of the committee to giving me the opportunity to present the project I'm doing in the lab of Romain Kozul and Aurel Piazza, which aims at understanding how the folding of the chromatin by the cohesin is affecting homology search during DNA repair. So as you know, double-strand break occurs on the DNA, as Emmanuel told you, and they can be repaired by the homologous recombination pathway, which is a universal pathway. This mechanism uses an homologous sequence present somewhere else in the genome. It is initiated by the resection that consists in the degradation of the double-stranded DNA to generate single-stranded DNA overhangs, thanks to several nucleases such as MRE11 and EXO1. Then this single-strand DNA is coated by Ra51 with the help of Ra52. And the nucleoprotein filament is a key factor to look for an homologous sequence. It will screen the genome to look for this homologous sequence. And it's important to keep in mind that this process is a dynamic between interrogation and reversion. So once this filament is stably associated with an homologous donor, the D-loop that is formed, which is the key product of homology search, can be extended by DNA synthesis and the repair will be completed to complete the repair. So in this project, we're mainly focusing on the homology search process. So what we know is that in vitro, it is influenced by the 3D conformation of the DNA. And in yeast, in vivo, it is influenced in the rubble genome organization. However, how is the spatial organization of the chromatin influencing homology search? And how the global genome organization is influenced in presence of damage, in presence of a damage is not well known. So we asked the two following questions. What is the consequence of a single double-strand break on global and local organization of the genome? And in turn, what is the influence of this organization on homology search? So to study genome organization, we are using the IC technology. So starting from a cellular culture, contacts between DNA are stabilized by the addition of formaldehyde. Then the DNA is digested by restriction enzymes, and DNA in proximity can be ligated during the ligation process, creating chimeric DNA. And the abundance of the chimeric DNA will be directly correlated with the frequency of contact. So after pair-end sequencing, alignment, and normalization, you end up with a contact map whose intensity is directly correlated with the frequency of contact. We can, from that, also extract what we call a distance law, which gives you the probability of contact in function of the distance. So the closer you are, the more likely you are to you have more chance to interact with the sequence, and conversely. So it was shown, thanks to this technique, that G1 population genome is mainly featureless with a flat distance law, which is not the case of metaphase chromosomes that are well individualized into cohesin arrays of loops, and that shows also an increase of contact within the range of 10 to 60 kb. So we are combining this technology with this experimental system that allows us to induce a single double-strand break by the expression of H2 endonuclease in presence of galactose. In this system, the cells are haploid, and the double-strand break is irreparable by homologous recombination. So most of the time, we start from an asynchronous culture. At T0, galactose is added to the media, so double-strand break is induced, and cells are collected 4 hours after double-strand break induction. At that time, 99% of the molecules have been cut. Based on the decrease of coverage, we know that 80% of the molecules have been resected. And based on D-loop formation quantification, we know that we have a high amount of D-loop formation here. So we know that D-loop invasion takes place and that we are in the middle of homology search. In addition, thanks to fact analysis, we realized that 4 hours after double-strand break, the cells population are mainly accumulated in G2M of the cell cycle. So this is completely consistent with the fact that when you have a damage in the cell, the damage is sensed and the checkpoint gets activated preventing cellular division upon damage and unrepaired DNA. So it's important to keep in mind that a majority of cells are undergoing homologous recombination in G2M arrested cells. Here is the contact map of the chromosome 5 before galactose addition. So as you can see, it is mainly featureless and mimics the structuration of a G1 population, which is consistent with the fact that this population is mainly enriched in G1. 4 hours after double-strand break induction, here is the structuration of the chromosome 5. So we observe 2 levels of reorganization, either locally in the vicinity of the break or globally that does not only concern the damaged chromosome. Locally in the vicinity of the break, so within the range of 50 kb, we observe that 2 border patterns that occur, two border patterns flanking the double-strand break site, and a perpendicular signal emanating from the break that may result from an increase of contact between both double-strand break ends. Globally, as the cells are accumulated in G2M of the cell cycle, so in metaphase due to checkpoint activation, they adopt the organization of metaphase chromosome. I mean by that they are well structured, well individualized, with an increase of contact within the range of 10 to 60 kb, and the distribution of the signal is made in triangles and dots, which is characteristic of an organization of array of loops. So metaphase chromosome structuration is well known now. So it was described recently that it is compacted into cohesin-mediated loops. So the cohesin is belonging to the SMC family. It is composed of 2 SMC subunits, NCMC1 and SMC3, and a third subunit, the cohesin SCC1. This complex is mostly known for its function in sister chromatid cohesion. Okay. However, it was shown that the cohesin-dependent loop in metaphase are mostly independent on sister chromatid cohesion. So we then wanted to validate that upon a damage, the genome was structured with the same determinant as a normal metaphase. So for that, we tested if this genome organization was dependent on cohesins. So we induced cohesin depletion with an auxin-inducible degrowth. So for that, the cohesin, the SCC1 is tagged with an AAD tag, and with auxin addition to the media, it is efficiently depleted. This depletion is induced at the same time as galactose addition to the media. 4 hours after double-strand break, in absence of cohesin, most of the structuration of the chromosome is lost. It is also observed that the genome has the same organization as the G1 genome. So we conclude that 4 hours after double-strand break, cells are mainly in G2M and they adopt a cohesin-dependent genome organization. However, locally, the structuration of the break is not influenced by SCC1 depletion, suggesting that it is independent on cohesin. So the impact, so we just wanted to check and confirm that the impact of cohesin on global genome organization was mostly independent on sister chromatid cohesion. And this is the case because when replication is prevented by the depletion of the replication factor CDC45, G1 release cells will still show a high level of structuration 4 hours after double-strand break induction. Just to clarify, when you have a double-strand break, either you have 1 chromatid or 2 chromatid, they still in both situations can highly structure their genome. So this shows that 4 hours after double-strand break induction, the sister chromatid cohesion only mainly contributes to genome folding. So we conclude that homologous recombination globally takes place in the context of metaphase chromosome and not in the context of featureless chromosome organization, which may somehow change our perception of how is homology search regulated. Now, what are the determinants of the local structure observed at the break site? So as I showed you before, it is independent on cohesin. And it may result from increase of contact between both double-strand break ends. As it is occurring at the break site, we wonder whether it was dependent on homologous recombination proteins and more especially the resection proteins, because the extent of this signal is following the extent of resection, as you can see by the decrease of the coverage in the vicinity of the break. So first, we tested if the MRX complex was involved in the structuration, because MRX complex is involved in resection, and it is also involved in the tethering of both double-strand break ends. In absence of AMR11 being part of this complex, resection is impaired, and most of the signal at the break has disappeared, which shows that the local organization of the break is dependent on the AMR complex. We then tested if the nuclease exo1, so nuclease activity of exo1 was required for this structuration. And as you can see in every exo1 nuclease dead mutant, the resection is strongly impaired and all the signal at the break has disappeared, suggesting that single-strand DNA generation by exo1 is required for this local interaction pattern. So the single-strand DNA allows the recruitment of RAF52 and RAF51 to form the nucleoprotein filaments. However, this local interaction pattern is not affected neither in RAF51 null mutant nor in RAF52 null mutant, suggesting that it is independent on HR proteins. The single-strand DNA is not only important for the nucleoprotein filament, but it is also important for checkpoint activation, as it recruits the 911 complex composed of DDC1, RAD17, and MEC3. This complex is important for the MEC1 activation, checkpoint activation pathway, and it is proposed to be mainly associated at the double-stranded, single-stranded DNA junction. Disrupting this complex by either deleting RAD17 or MEC3 led to the complete disappearance of the local interaction pattern on the break site without affecting the resection rate. So this is the scenario, the model that we propose from this data. So soon after double-strand break is induced, the MRX complex is recruited to tether both double-strand break ends. Then the MRX favors the recruitment of exo1, a nuclease that will extensively degrade double-strand break ends, and redistribute the 911 clamp at the double-stranded, single-stranded DNA junction. This clamp has the ability to dimerize through RAD17. So we propose that it could be structurally involved in the bridging of both double-strand break ends. So our interpretation of this signal is actually, so for each double-strand break, the double-strand break ends are bridged together at the double-stranded, single-stranded DNA junction, so at one point by the 911 complex. And because of the heterogeneity of the resection at the cell population level, we get this broad perpendicular signal. So now the question we ask is, how is the global genome organization influencing homology search process? So we first wanted to know with IC contact data if we could detect contact made by a double-strand break site with its inter-homologous donor. So for that, we are zooming on the contact map and we focus our attention on the double-strand break site and the inter-homologous donor. So this is the amount of contact we detect 4 hours after double-strand break induction. These contacts are dependent on homology because when the donor is deleted, so the homology is deleted, 2/3 of the signal is lost. It is also dependent on the nucleoprotein filament because in absence of RAF52 or RAF51, 75% of the signal is lost. And conversely, D loops are destabilized by SGS1 on helicase that will then destabilize the contact made by a break with its homologous donor. And in absence of SGS1, we observe an increase of 2-fold of the contact made by the break and its homologous donor. So they are dependent on homology and homologous recombination factors. So we conclude that we can capture contact made by the double-strand break and its inter-donor region during homology search. Now, can we detect contact but genome-wide? So because IC technology can only detect contacts between double-stranded DNA, we cannot directly look at the nucleoprotein filament. So we decided to focus our attention on a region located at 7 kb from the break site, long of 10 kb, and being partially resected. So we can either look at the left or the right of the break. So for example, we can look at the break at the left viewpoint. So we wondered what is this region contacting 4 hours after the double-strand break in comparison to a normal metaphase. So we are selecting this stripe of contact, not only on the chromosome 5, but also on other chromosomes. These stripes are then subsampled, and the ratio between them is performed by serpentine that allows us to decrease the noise and dynamically adjust the binning. And we end up with a stripe of contact that concerns, for example, the left side of the break. In red, you observe the contacts that are preferentially made 4 hours after the double-strand break with all the chromosomes. And in blue, the contacts that are preferentially observed in a non-damaged metaphase. So based on the increase of the red signal, you can see that the flanking regions of the break are more contacting the inter-chromosomal sequences 4 hours after double-strand break induction. In cis, which represent the majority of the contacts, you can see that the exploration is limited to the vicinity of the break and done in site-specific way. I mean by that the left is more contacting the left and the right more the right. The increase of contacts made by the double-strand break and the inter-chromosomal sequences seems to be dependent on RAF51, because in absence of RAF51, 4 hours after double-strand break, we don't observe such an increase of contact. We don't observe so much red signal. This is not the case for the cis contacts, as if the exploration in cis was depending on another mechanisms. So we can use this approach to visualize homology search genome-wide. So the question we had is global genome organization influencing homology search? So to answer this question, we looked at the distribution of contacts made in absence of cohesin and in presence of cohesin 4 hours after double-strand break. As you can see, we observe a huge increase in the red signal, suggesting that the double-strand break ends are more contacting inter-chromosomal sequences in absence of cohesin. And also in cis, we observe a huge increase in the red signal, suggesting that there is also an increase of contact, but at a longer range distances. And we also lose the site-specific way of homology search. Consistently with the increase of contact of the break with the inter-chromosomal sequences, we observe an increase of contact with the break site with its homologous donor in absence of cohesins. So these results show that cohesins tends to inhibit genome-wide homology search and promote intra-chromosomal and site-specific homology search. This regulation by the cohesin, for the second time, I'm not showing you the data, but this regulation of the homology search by cohesin is mainly independent on sister chromatid cohesion. So because with IC technology, we cannot directly look at the nucleoprotein filament, we decided to confirm our data so that the homology search is regulated by cohesin by looking directly at the D-loop using a D-loop capture assay. So this is the amount of D-loops that we can detect 4 hours after double-strand break induction between the double-strand break site at the inter-homologous donor. This level of D-loop is decreased when an intra-chromosomic donor long of 0.6 kb is added. And this decrease is even more stronger when the intra-chromosomic donor added is longer. So this suggests that intra-chromosomic sequences are favored and kinetically advantaged for D-loop formation over inter-chromosomic sequences. So as cohesins tend to restrict homology search intra-chromosomically, we wonder whether this preference could be dependent on cohesin. So to test this hypothesis, we looked at the impact of cohesin depletion on this ratio, so the ratio of the amount of D-loop detected intra-chromosomically over the amount of D-loop detected inter-chromosomically. So in peak, you can see the D-loop, the ratio that we get in the control. And in absence of cohesin, this ratio is decreased, suggesting that less D-loop are detected intra-chromosomically and more D-loops are detected on the inter-chromosomal sequence, which is completely consistent with the data that we got from the contact, from the IC, that cohesins tends to restrict the homology search intra-chromosomically and disfavor homology search in inter. So our working model is the following. So homologous recombination and homology search is mainly occurring in the context of metaphase or organized genome, where the homology search is restricted intra-chromosomically and mostly by the cohesin, and to disfavor the contact with the inter-chromosomal sequences and therefore limit gross genome rearrangement. And this effect is mainly done by the cohesin and mainly independent on sister chromatid cohesion. So I would like to thank the people from the RSG and the MG Lab, and especially the contributor of this project. So Romain Kozoul, Fabien Girard, Agnès Thierry. and Aurel Piazza, of course, Jérôme Savoco, and Agnès Dumont. I also would like to thank Charlotte and Charlie Cochran and Brenna to help me for the repetition of this presentation, and thank you for your attention.
Merci, merci beaucoup. Donc, on a une dizaine de minutes pour les questions. Karine, on va commencer avec Karine Dubrana.
Ouais.
Oui, alors désolé, en fait, j'avais oublié de baisser la main, mais j'ai quand même une question.
Ok. Bon, j'ai été la première. Okay.
Il n'y a pas de souci. Par rapport au modèle que vous faites, est-ce que vous faites un lien entre la structure que vous voyez et les contacts que vous pouvez voir d'une extrémité avec les séquences adjacentes? Est-ce que vous arrivez à faire un lien dans les différents mutants qui font disparaître cette fameuse structure perpendiculaire en C?
En fait, je ne suis pas sûre de comprendre la question. Est-ce que dans...
Si vous regardez l'orientation des brins, des contacts d'un côté de la cassure avec le reste du génome,
okay.
de la cassure avec le reste du génome, vous voyez des changements dans les différents mutants qui font disparaître ce fameux spike.
Oui, on augmente. Alors, il me semble que quand on empêche le contact entre les deux extrémités de la cassure, on augmente les contacts d'un bras avec l'autre. Enfin, d'un côté avec l'autre. En fait, le bras droit va être plus mobile et va pouvoir davantage explorer le côté gauche de la cassure et inversement.
Donc, notamment dans un mutant RAD17, vous voyez ça?
Oui.
D'accord. Merci. Et c'était super.
Alors, une autre, Emmanuel, tu as levé la main.
Oui, Hélène, j'ai peut-être raté ça, mais vous avez déplacé la cassure à différents endroits des chromosomes et est-ce qu'à proximité du centromère, qu'est-ce que tu vois?
Alors, donc on l'a fait, donc pour la première question, on a également mis la cassure dans le milieu à peu près du chromosome 4, mais ça je ne l'ai pas présenté, et on observe les deux mêmes niveaux de réorganisation du génome. Et alors, pour le centromère, peut-être que c'est plus simple si je repartage mon écran. En fait, ce qu'on s'aperçoit, c'est que là, sur le chromosome 5, la cassure est très proche du centromère. Notamment, le côté droit est très proche du centromère et on voit qu'il est assez contraint. Je ne sais plus, je crois 30 kb. 30 kb peut-être. Je ne me rappelle plus très bien. Ouais, je crois que c'est à peu près ça. 30 à 50, entre 30 et 50 kb. Donc, il est très contraint. En fait, on voit qu'il fait beaucoup moins de contact avec le reste du génome que ne le fait le bras gauche, qui est moins contraint par sa proximité du télomère.
Et tu le vois interagir avec les autres centromères?
Voilà, exactement. Ouais. Avec les autres centromères, oui, sûrement. Enfin, il me semble que oui. Je suis pas sûre, mais il me semble que oui.
Ce serait intéressant de comparer nos données parce qu'on a aussi tout un aspect que je n'ai pas présenté aujourd'hui sur les cassures le long des chromosomes et comment on se fait la recherche d'homologie. Alors, on n'a pas vos outils, mais ce serait intéressant de vraiment croiser nos deux données.
Oui, ouais, tout à fait.
Ok, Angela.
Oui, merci Hélène, c'était super. Donc, j'ai une question par rapport à cette histoire de recherche préférentielle à gauche et à droite. Vous m'entendez?
Oui.
Ok. Parce que, en fait, est-ce qu'on est vraiment sûr que le simple brin contacte préférentiellement de gauche et de droite? J'ai dit, c'est en fait des interactions entre les deux brins. Donc, je me demandais si c'était ce que tu voyais, c'était pas juste le fait que la cassure est bien sûr un obstacle à l'extrusion de la boucle par les cohésines et que donc forcément tu fais des boucles à gauche, tu fais des boucles à droite, donc c'est pour ça que tu vois plus de contacts de part et d'autre, mais ça te dit pas forcément, enfin je voudrais savoir si ça te dit que vraiment que le simple brin lui-même de gauche a envahi plus facilement à gauche et celui de droite à droite. Je sais pas si c'est clair, mais...
Donc pour vraiment,
est-ce que par exemple si tu fais un essai de 10 loop capture avec un donneur à gauche ou à droite, tu as une différence?
Exactement. Mais en fait, du coup, pour vraiment répondre à cette question-là, parce qu'à cause du IC, on peut regarder que le double brin, il faudrait qu'on fasse ça en fait dans le système. C'est ce qu'on prévoit faire, enfin plutôt dans le chromosome 4 où là on a plus de mobilité des deux extrémités de la cassure. de pouvoir mettre une cassette éventuellement des deux côtés pour pouvoir tester ça, parce que là, sinon, on ne peut pas le voir sur la base des données d'IC. Et par rapport à ce mécanisme d'expansion de boucles, c'est possible, mais en fait, comme la taille des boucles, elle est de 10 à 20 kb, en fait, la recherche d'homologie, elle va du coup être assez limitée en fait, du côté gauche ou du côté droit, ce qui ne correspond pas tout à fait aux échelles de grandeur qu'on a de contact, soit de la gauche ou de la droite. On a plutôt une échelle de 60 kb. Donc, le simple mécanisme de loop expansion ne peut pas expliquer ce contact préférentiel, cette spécifique gauche et gauche et droite et droite.
Ouais, je sais pas. C'est pas évident parce que c'est le fait que c'est pas seulement la taille de la boucle qui fait ça, c'est le fait que tu puisses pas passer de l'autre côté qui fait que forcément ça te fait un effet différentiel. Enfin, je sais pas, faudrait modéliser. Ça me paraît pas si intuitif que ça qu'on puisse conclure juste comme ça, mais je pense que la D-loop capture, ça te donnera la réponse de toute façon.
Ouais, complètement.
Ok.
Merci.
Merci. Est-ce qu'il y a encore des questions? Apparemment non. Alors, on va peut-être, pour rattraper un peu le retard, on va passer à la dernière communication.
Ok, merci.
Merci.